We've got one vote for number 3 being the best. Why do you think that number 3 was the best answer?
I just summed up the amount of T's and A's, and then I divided them by 20.
Okay, so we've got 3 A's out of 20 nucleotides, which is
Around 15%.
15%. Okay. Did they all meet that criteria? Are any of these answers wrong? Comments? What do you think?
None of the answers are wrong, but a couple of them have like decimal answers, not real numbers. So you had to kind of like round up or down.
Sure. So which ones had decimal answers?
Like 1, 2.
Yeah, 1 and 2 are identical.
5.
Someone was collaborating. Not cheating, collaborating. Right. So yeah, 5 A's out of 34 nucleotides isn't quite 15%, it's 14 point something. But those are right. Are those answers, are 1 and 2 better or worse than 6? How did answer number 6 come about, do you think? It's 100 letters long, it's 100 base pairs, A's, T's, G's, and C's. So how many A's are there? 15. And there's going to be 15 T's and 35 C's and 35 G's. Right? That's the fast way. That's right. That's the fast way to do it. You don't have to do any math. It takes probably a lot longer to type them out and make sure you've typed the right number of letters. But that's right. So all of these, in their own way, are acceptable answers. Why am I making this point? Don't assume that there's always just one right answer. There were lots of different answers I received, which is one of the reasons I'm not grading this. But there were lots of right answers. They all looked different. If I had any complaint about any of this, it would be the commas, which isn't standard nucleic acid sequence annotation, but it's fine for this case. Does it matter if it's lowercase versus uppercase, 4 versus the others? Not to me. So in general, everybody did a really good job on answering this question. Simple question. Create a DNA sequence to do something that has certain characteristics. So this is what we're going to work on today. We've just done step 1, reviewed what we did last class. I'm going to stick in, so everything in blue is the typical class structure. So we will always come in, there will always be a, while you're waiting, something to practice if you like. The very first thing I will do after that, after we review the answers to whatever we discussed last class, I'm going to ask, does anybody have any questions? This is when I would love for you to take over. I come to class every day with 50 minutes worth of stuff to say and do. I prefer answering questions because that helps me make sure that you're getting the support you need in learning what it is you need to learn. So first, review. Second, I'm going to ask, does anybody have any questions? Then there will be pin drop and you can hear it. But by the end of the term, everyone's going to come with questions. That's the goal. Okay, we haven't really started getting into the topic and the content yet, so I understand if nobody has any questions today. I'll ask in just a minute. Then for the start of every chapter, I'm going to go through core concepts and vocabulary. So that's to give you an idea of what I'm expecting you to learn from each section of each chapter. One slide, just here's the basics, what you need to know. We'll do a Socrative exercise, a quiz, or a poll to make sure that you understand the material to be covered in class today. Then we move on to the bulk of the section, the bulk of our class meeting, exercises and discussion. I prepare something for us to do in class. We do it together. We discuss the results. Those 2 red bullet points, I'm pointing out to you specifically, this is how you learn how I write exam questions. That plus the while you're waiting, the pre-class exercise. All of this should give you an idea of what I'm going to do on exams. So please feel free to ask me later, but if you ask me, so what's going to be on the exam, or how should I study for the exam, I'm going to say, remember all that stuff we did during class? That was all preparation for the exam. So feel free to ask me again later, but that's what I'm going to tell you later. Study the lecture slides, study the exercises we did, study the quizzes that we did on Socrative, etc. Then at the close, we'll talk about what's going to happen for the next class, homework that's due, et cetera. And we'll close with some sort of short summary, minute paper sometimes. We'll do that today. So one of the questions from Socrative on day one was, what is it that you think you're going to learn about or you're interested in learning about in genetics? Here are the answers in word cloud format. You're going to learn and think about genetics. I don't know if that's particularly creative, but what else stands out here? Does anybody see any other words that resonate with you? What do you all think you're going to learn about this term? We've got mutations, genes, diseases was something, so most of you said, hey, maybe we'll learn about diseases. So I summarized all of those answers, and this is, again, by way of thinking about the broad structure of the class across the term. This is basically what people said. There were some detailed, one person said this or one person said that, but these were broad themes that stood out to me. People are interested in how DNA mutates or changes, how variation exists, so why we all have different skin color, hair texture, what have you. A lot of people said genetically modified organisms, which is no surprise. We're in a highly agricultural area. You want to know about how DNA technology can be applied to life. A few of you said you want to know how a cell knows which of our genes to turn on at any one point in time, which is a really advanced question. I'm glad those of you that said it did. I'm impressed. We'll cover that partly here, and if you're really interested in that topic, you should also think about taking Biology 155, Developmental Biology. So altogether, many of you indicated that you wanted to learn about what we're starting the term with, molecular genetics. So those are all questions that molecular geneticists ask. So there are basically three different subdisciplines, big subdisciplines of genetics. Those are molecular genetics that deal with the molecule of genetics, DNA. A lot of you indicated that you wanted to learn about inheritance patterns. People that might be interested in breeding design, crosses, getting desired traits in organisms. That defines a field called, if I could draw S's a little bit better, transmission genetics. So that's the second broad topic that we're going to talk about in class this term. And then the last batch, why do we all have different eye color, skin color? We'll talk a lot about earwax texture. You probably weren't aware that humans basically have one of two types of earwax. There's wet and there's dry. We'll talk about that. That's an example of a phenotypic difference. Evolution and disease. All of this has a particular theme. We start with mutation, how DNA sequences change, how our DNA changes over time. And we wind up at the bottom with phenotypic differences. So we go from genotype to phenotype. What is that process? What do we call that? DNA to RNA to protein is transcription followed by translation. And that whole thing where DNA makes RNA makes protein is called the central dogma. So how changes in our DNA lead to changes in the proteins we produce and how we look, how we act. So that's how the course is structured. We start looking at the molecule of DNA itself. Then we start looking at transmission genetics, how that molecule is passed from generation to generation. And then at the end of the term, we talk about how DNA differences between us cause the phenotypic or visual differences between us and lots of other organisms. One person said what I was expecting all of you to say, this point down there in the bottom right. I did not want to learn about Punnett squares and probability and statistics. Now, normally I like creating a really safe, comfortable atmosphere to share your opinions, but I think this might be one where everyone's okay raising their hands. How many of you agree with that sentiment? You didn't necessarily, okay, so about half the class or so. Okay, so unfortunately, I'm here to say that we are going to do Punnett squares, we are going to do probability, but I understand that this is a major barrier for a lot of people in their enjoyment of genetics, if there is one. And so we're going to treat those really carefully and thoughtfully as we go through the semester. Okay. Last note about class structure. The exercises that we do in class are for you to get feedback for yourself about how much you understand and have learned the material. And so if you're not here, especially those of you in the back, if you're not here, I don't know, it just tends to be that people in the back sometimes tend to come in, have been the ones that came in late or leave early. If you're not here participating, you don't get that benefit of getting feedback for yourself about how well you understand the material. A lot of what we've been doing, the Google Classroom exercises, the Socrative quizzes, those are more for you than for me. The first week I use them to take attendance so I can kick you out if you don't come. I don't want to do that, but since there's a wait list, I need to do that. After that, all of these exercises are really for your benefit, to get understanding about whether or not you understand material that's going to show up on the exams. And if you're not here and you don't participate, you don't get that benefit. Bottom line for me, what I prefer to see is everybody get an A. We already talked last class about the fact that I don't curve. I would love for everybody to get an A. There is not a certain percentage of you that are going to fail because I say so. I want everybody to succeed, and most of all, I want people to demonstrate growth. Meaning that if you're doing really kick-ass at the end of the semester, it might not matter so much that you started off feeling insecure or that you did poorly on the first exam. Right? We throw out an exam grade. That's your opportunity to learn about how I teach and how I write exams. So among all of, these are all tips for succeeding in my class the way I teach and the way the class is designed. The most important thing is to show growth. Doesn't matter how you start the semester, it matters how you finish. Blah, blah, blah. Oh, sorry, guys. Now, hopefully everybody's read chapter 9. We started with chapter 9 because this is molecular genetics. That's where it is in this textbook. So don't freak out that I didn't start with chapter 1. We'll be okay, I think. But this is molecular genetics. Chapter 9 is the first chapter. Does anybody have any questions about the reading that was to be done for today about this chapter? Pin drop. Crickets. Be creative. Anything that you felt like you didn't understand or you want more information about?
So is it okay just to read the highlights, or should we read the whole thing?
Great question.
Highlighted.
So SmartBook comes with items that are already highlighted. Should you read just those? Do you have to read the whole chapter? Or at least the sections that I assigned of the chapter? My advice is, let's see. My advice is to start, if you don't have time, look, we're all adults. I'm probably more adult because I've been around on the planet longer than many of you. But we're all, honestly, this is my opinion about us and our interaction. You have a lot of stuff on your plate. I've taught this class before. I make lots of new material every time I teach this class, so I invest a lot in this class. And you've got a lot on your plate. So you have to make priorities about what you're going to spend your time doing. And yeah, absolutely, be efficient. If you don't have time to read the whole chapter, just do the highlights. Come to class, like we're going to do today, see what we're going to do in class about the chapter 9 material. Participate in the exercises. That'll get you most of the way to what you need to know about what the chapter 9 material is. So start with the highlights. If you've got more time that you want to devote to reading, then start going in more detail. Is that reasonable? Yeah. Other questions?
Dr. Rock?
Yep.
So, I know you sent out an email about this, but for the 2 apps, I don't know if it might not be a question for you. Which one's the one where we don't get graded, or we can use to study? Which one's the one where we do get graded?
Oh, the 2 apps.
Yes.
Has anybody figured this out yet? Which of the 2 apps do you can actually do? I don't. So Learn Smart. you connect to the class through the app, and then you can do the assignment. We're going to learn soon whether or not that actually passes your grades from the McGraw-Hill website to Blackboard. But that's my issue, that's not your issue. Any comments or clarifications about the apps at this point? Here's some discussion. Yes. Yes. That's right. So this is the whole point of using, you can definitely use the apps to study. The best way to ensure that your grades are going to pass from the McGraw-Hill website where you actually do the book access and the studying, the Learn Smart reading, that website sends your grades back to Blackboard's Grade Center. If you access the course assignment through the link that I provide in Blackboard. This is the, if you want to be my friend this semester, please, 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 always start by clicking the link in Blackboard, which will take you to McGraw-Hill's website, and make sure that your grades get sent back to Grade Center. If you don't do that, the McGraw-Hill website should still record what you've done at the McGraw-Hill website. It will show up in your Grade Center, whatever it's called, at the publisher website. It just doesn't send it back to Blackboard. So then you email me a week from the end of the term, you say, Dr. Ross, I swear I did these, but September 15th, October 24th, and 11/13 aren't showing up in Grade Center. I swear I did them, and then I go to the McGraw-Hill website and I have to manually enter those grades in Grade Center so you can see them. So this is not an essential thing, it's a please help me help you understand where you sit grade-wise. Go to the link in Blackboard first to the McGraw-Hill website. Otherwise it's a lot of manual entry, which I'm happy to do, but I prefer not to if I don't have to. Other questions? Course Mechanics, Chapter 9 material. What was Chapter 9 about? DNA. So molecular genetics. We're starting with what the hell is DNA? So here I told you every chapter or every group of sections for every class period, I'll tell you what I think are the highlights. What should you be getting out of this experience? So I'd like everybody to be comfortable with the structure of a nucleotide, the structure of a strand of nucleotides, what a double helix looks like. We're going to practice today Chargaff's rule. We started looking at his data last class. That's what our exercise today is on. So that's obviously going to be a focus of your ability to apply information on an exam. And Chargaff's rule is related to base pairing and complementation. We'll talk about hydrogen bonding and anti-parallel orientation and the differences between DNA and RNA. Those are the core concepts for me for this chapter. At the end of the class today, when we do that 1-minute paper where I ask you to summarize what you think, I'll ask you two things. Summarize what you think you learned today, and I'm going to ask you what was the one thing or few things that you thought were least clear about today, or items that you now have questions about based on what we did in class. This is a really great point for you to give me feedback on whether or not any of you individually feel uncomfortable with any of the terms we've been using in class, because genetics is laden with all of this vocabulary, as are all scientific disciplines. So I don't define these in class because they're defined in the textbook, which you've read for class today. But when you feel particularly uncomfortable or uneasy or unsure about limitations to vocabulary or something like that, please let me know in those end of class feedbacks, if you don't ask during class. So here's the part of class where we go to Socrative and do a little quiz. This one won't be scored. This is just for your personal feedback, but please do your best on concepts for today. So it's a short multiple choice quiz. Please take it.
Dr. Ross?
Yep.
I'm not able to connect to it.
Okay. So, and again, this is. Okay. Is anybody else having internet connection issues? Uh-oh.
Might want to restart your tablet.
I already did.
Thank you for the text.
What Wi-Fi are you on?
It's not showing that.
Yeah, it's not even showing the networks yet. It's still looking for that. for those of you that are having those issues, again, I'm not scoring it, but you should be able to get the benefit of getting feedback on your understanding. So, let's do this. For those of you that don't have internet access, I'll show you the questions. And feel free to write them down on paper, make digital notes on your tablets. Well, I can show you part of it anyway. Do those questions at least.
Can you show number 4?
I would be happy to scroll a little bit, just for a second. So this picture. There's a picture from the previous question. Is that DNA or RNA? How can you tell? And do absolutely talk to each other. This is about learning. This is not a quiz. This is for your feedback. If you want help, go for it.
Yeah, CC, yeah.
It keeps showing the one we have to write it doesn't show this.
I haven't found the right one.
Oh, so you submitted it.
I'm all like trying to like.
He's like. She's like
Okay, so we're almost done. Okay. So let's pause at least. You can submit your answers at any point if you want, but let's talk. You might not have made it all the way through, but maybe you did. Okay. So if you have this DNA strand whose sequence is given, so if the DNA strand is 5' TAG, it's my favorite DNA sequence, the only one I can figure out that spells a sentence. Tag a cat. There are not very many vowels in DNA, people. You've got to be creative. So tag a cat. What is the sequence of the complementary strand? So this is one strand of DNA. It's base paired with the complementary strand. So what's the sequence of that strand? T's pair with A's. A's pair with T's, A's pair with T's, T's pair with A's. What about G's and C's? Right? Just review, hopefully. Okay. So that should be the sequence of the complementary strand. In the first strand, every T is paired with an A, every G is paired with a C. What do I have to do to complete this? I have to do the 3' and the 5'. Sir, do you have a question? Okay, that's okay. That's all right. So I have to do the 5', 3' thing on the bottom strand. So this is an example of complementation. A's pair with T's and G's pair with C's. And then the polarity has to do with this notation, I, T, Y. So what's the A? Which end of that strand is it? 3' or 5'? 3'. Very good. Right. Which means the other end of this DNA strand is 5'. And that's because DNA is anti-parallel. So if DNA is double-stranded, you'll always find one end's 3' end next to, adjacent to, the 5' end of the other strand. So hopefully all of this is review. I'm not trying to say that this is anything you haven't heard before. I just want to make sure that everyone's comfortable. This is where we're starting genetics. Everybody has this information. A's pair with T's, G's pair with C's. DNA strands are anti-parallel. Questions at this point? Yeah.
So, I did actually get a little bit confused.
Sure.
When it says 5' and 3', they're talking about, is that the sugar molecule? They're talking about the 135 thing and everything else?
Right, so we're talking about, I did not pay him to ask that question, but nice lead in. So how do we determine what the orientation of the DNA molecule is? I mean, it's fine to say there's 5' and 3', but how do we figure that out on our own? Why is it important to talk about 5' and 3' in the first place? What does this say? Imagine wo nig sufnock it dlo beef. Right, what's the point? We have to have, yeah? Say that louder.
Well, you wouldn't be able to read it. I don't know what direction.
You wouldn't be able to read it if you didn't know what direction you were supposed to read it in. So if we had no convention, I'm talking about Western cultures now, just for a second. We have a convention, hopefully all of you appreciate, that we write from left to right, and we read from left to right. If we didn't all agree that that was how we were going to do things, then it would take you a lot longer to read certain passages. So by the same token, geneticists have decided that we're going to always read DNA sequences from 5' to 3' so that we can all agree on what the word, the DNA sequence, is, what it means. Okay. Okay. So which end, this is one of the questions, which end of this, which direction do we read this DNA word? Is it CGAT? Or is it TAGC? Let's do a show of hands for CGAT. Who thinks that that's the right answer? So we've got like 2/3 of the class. What about TAGC? Uh-oh. See, this is why I like Socrative, because the answers to the first poll don't bias the answers to the second poll because everyone's anonymous. Nobody knows what everybody else is saying. Okay, so the majority of you said it's CGAT. The question I will always ask you after you answer, why? Why is that the answer? Because it's drawn like that? Because we go left to right, top to bottom in Western culture, and that's the way it goes. So it's the, go ahead, sir, it's the orientation of the molecules.
I think it's going from top to bottom because the phosphodiester link always connects at the 5' carbon and the base always connects at the 1' carbon.
So it's all about the chemistry that you had before you came to this class. Numbering of carbon rings. Oh, and I was an organic chemist too. So the phosphate groups on every nucleotide, this is why studying nucleotide structure is important. I'm going to pick somebody at random. No, I'm not going to pick somebody at random. That would just be mean, wouldn't it? Who feels comfortable describing what comprises a nucleotide up here? Somebody want to? Yes, sir. Here. Use your finger, circle one nucleotide in that. Fabulous. All right. Thank you, sir. Now it's not just me doing the teaching. Farming out the labor. Okay. So how many nucleotides are there in this molecule? There are 4 units, 4 nucleotides. You call it a tetranucleotide if you want to, 4 nucleotides. So each nucleotide has that sugar, that ring, sugar. I don't know why my S's don't work with the stylus. Okay, so the phosphate is connected to the 5' carbon. Where's the next nucleotide connected? Which carbon is that one? That's the 3' carbon. You don't have to know why, you just have to know that the phosphate's connected to the 5'. I won't make you do that. So every nucleotide goes 1 phosphate, 2 5' carbon, 3, just in order. The 1, 2, and 3 doesn't matter, it's just arbitrary. I could say A, B, and C. First it's the phosphate, then the 5' carbon, then the 3' carbon, and then on the very last nucleotide, what's connected to the 3' carbon? OH is also called hydroxyl. So there's a hydroxyl OH on every 3' carbon. Phosphate on every 5' carbon, hydroxyl on every 3' carbon. So that's how we define the orientation of this molecule. We say, okay, what, let's see, switch colors again. There's the nucleotide, or at least the base. There's a C, there's a G, there's an A, there's a T. You don't have to memorize the structures. I will never ask you, probably. Here's a nucleotide, what base is it? Is it A, C, G, or T? So that one is at the 5' end. Here's the free 5' nucleotide. It's the one with the 5' phosphate that's hanging out there, unattached to anything else. 5' end. 3' end has that free hydroxyl group on the 3' carbon. So to summarize, any DNA sequence has a 5' phosphate. Then you would write the nucleotide sequence. And then that very last nucleotide will have a free 3' hydroxide, yeah. So that's how we get that 5' to 3' nomenclature. If you wanted to, I could give you any strand of DNA structure like this. All you'd have to do is figure out where's that free phosphate, that's the 5' end, where's the OH, that's the 3' end. Questions, concerns, clarifications?
Actually, yeah, so while we're still on this slide.
Yeah.
Where would so in McGraw-Hill, the LearnSmart thing, they're talking about on the carbon atom, the 1, 3, and 5, that's kind of more what I was confused on. I didn't understand what they were talking about, where the phosphate, the sugar, and then the nucleotide base all connected.
Right. So that's partly what we're talking about here, but come up after class, show me an example of what you're talking about, and we'll work through it.
All right.
Okay? Excellent. But yeah, please do. So that's all organic chemistry. This is not an organic chemistry class. So is this RNA or DNA? Who says DNA? Anybody want to take RNA? Why? Oh, I didn't think about that. Right. That was not the answer I was looking for, but you're totally right. Which is why you should always question people, because sometimes they have these expectations that are unclear. So thymine, the base that we abbreviate T, is only found in DNA. What is it in RNA? Uracil. So it would be U if it was RNA. Okay, what else tells us that this is not RNA?
I think it's in the name, because DNA dehydroxy, so it doesn't have another OH at the 2' position, which RNA does have it, it's called ribose.
Right. So if this was ribonucleic acid, it would have a hydroxyl group on the carbon there. If it's deoxy ribonucleic acid, it has D, or one fewer oxys, hydroxys, so it doesn't have that OH group. Good. Okay. Other questions about this? About any of the questions on that Socrative quiz? Let's talk quickly about base pairing. Does anybody have any questions on this topic? What's the most important thing to know that's illustrated up here about base pairing? Again, this should be reviewed, but let's make sure we're all on the same page. G's pair with C's, A's pair with T's. What other information is in these figures? Right. How many hydrogen bonds? How many hydrogen bonds are there? They're the dashed lines. So G's and C's make 3. A's and T's make 2. Why does this matter? Let's say we live in a time of climate change. If you don't think that that's true, then let's just hypothesize that we're in a time of climate change. The world's getting warmer. It's affecting species. Would you rather have a genome that has sets of chromosomes? Would you rather your DNA have lots of G's and C's in it, or more A's and T's? Wow. There's more. So there was a lot of G's and C's there. I'm impressed. There are more hydrogen bonds. So what does that do to the DNA if there are more hydrogen bonds between the two strands? It's more stable. It's harder to break all of those bonds that are holding the two strands together. Which tells us two things about DNA that are important. One is that DNA being double-stranded, the two strands being hydrogen bonded to each other, is really important. And it tells us that temperature affects different DNA strands, different DNA duplexes, double-stranded DNA, in different ways. Sir?
Wouldn't that also affect like, what do you call it, when the double strands, kind of try to like replicate themselves? Would that
So-
also affect it?
Yeah. So when do we want our DNA strands to come apart? Replication. That's how we replicate our DNA. They have to peel, the enzymes have to peel those two strands of DNA apart to make copies of it. So maybe it's not so good to have lots of G's and C's in your DNA because maybe that makes it more difficult for those enzymes to pry those two strands of DNA apart. So now everybody maybe should be thinking to themselves, is there an optimal amount of G's and C's and A's and T's in the genome so that you balance the propensity for DNA to just melt, become single-stranded by itself given environmental temperature, versus the fact that we need enzymes to be able to separate the DNA to replicate it? That would be a good question to ask yourself. Okay. So I prepared this fabulous thing, but like I said, I'm trying to practice what I preach. We'll see if I succeed. I prepared an exercise in a Google Sheet for playing with Chargaff's data. And I would like you all to work on this before next class. And I wanted to have a little bit of time in class to walk through the first couple of steps with you, and I will do that. I want you to know that I also made a video yesterday, and I've uploaded that to the YouTube channel, of how to do this Google Sheet analysis. I've started you off in this video. So if we don't get far enough in class today, you should be able to look at the video. And again, this is not for turning in for points. It's just for you practicing a DNA sequence analysis, playing with Chargaff's data. So let me turn to that Google Sheet.